Good morning. I'm back with your next class in histology. I hope my voice is clear and that all of you are attending regularly because we will soon be starting the gross anatomy of the abdomen. We're moving beyond thorax, so the next part will start, and you have to correlate what is happening in gross anatomy with what is happening in histology and parallelly in physiology and biochemistry. So in the last class we had looked at histology of the stomach, and we had said there was a cardiac end, there was a fundus, a body, and a pyloric region which had a pyloric antrum and a canal which ends in the pyloric sphincter. And depending on the function of each part of the stomach, its lining had changed in microstructure to subserve its function of that area. And we looked at two major slides. One was of the body, similar to the fundus, and one was of the pyloric region, and they had two different kinds of structure. Now we'll move on to the next part of the gut, which is the small intestine, where the major function now is absorption of the already begun to be digested food. In the stomach, the component which was getting digested, almost there, is protein. And in the small intestine, the parts which will continue to get digested are polysaccharides, the carbohydrates which had started digestion in the mouth with the salivary gland secretions, and all the remaining parts of the carbohydrates will also start getting digested in this region. Plus the new component called lipids will also start getting emulsified and digested in the small intestine and absorbed almost immediately and simultaneously. So the lining of the small intestine has to be adapted to both secrete items such as enzymes for the digestion of the contents of the small intestine, as well as some modifications of the surface which will allow it to absorb in large quantities all these digested products into the bloodstream and into the lymphatic capillary network. So we will look at the microstructure from that point of view. What we do know is that your intestines are divided into small and large, and in this, the small intestine is a very long piece of gut. There's about a quarter meter, about 25 centimeters of duodenum, then about 2 meters of jejunum and 3 meters of ileum. So these three components together, duodenum plus jejunum plus ileum, together make up what we are calling the small intestine. Their architecture is similar throughout, but there are important differences between the three. And in your practicals, you will see 3 slides which describe these 3 places. They look very similar, but at the same time, the discerning eye, the trained eye, will be able to make out the differences, and you'll be able to identify and to discuss correctly. So if you looked at it diagrammatically, the small intestine has this kind of a look with some surface projections, finger-like projections from this, if you can call it the surface. Actually, this is the surface. If we put a line through at this point, this would be the surface, above which some projections are going up, finger-like processes called villi, and below which some pockets or in-pocketings or pits are available, which are the intestinal glands. This is called the intestinal gland. I hope you can read it here. So if there is, you don't see anything like a level. There is no way of seeing anything like a level, either with the naked eye or with even in a section. There is no particular feature characteristic where there is a line saying this is the surface and above this is the villus and below this is the gland. So it's just by the structure of the wavy continuity of the epithelium and the lining cells of each part that you can differentiate. But for that you need a high magnification. We will look at that later. So what essentially we are seeing is the same 4 areas of the wall of the tube. We had an outer serosa, then you had a muscularis externa, then you had a submucosa, then you had a muscularis mucosae, then you had the lamina propria, then you had the lining epithelium of the internal mucosa. So we have epithelial cells. All of these are epithelial cells. Here they are only shown as one single line of square looking cells with dots, but those are the ones which have sophisticated changes where the composition of those lining cells will determine the job, the work that is done by the gut. We also supposed to have goblet cells, intestinal glands, which are also called the crypts of Leberkuhn. You might have heard of that term before. And we will have in the inner wall, in the mucosa, structures called microvilli, projections of the surface, inside which, in the core of which of each of these projections is a microcapillary network, which are fenestrated capillaries. Inside the lamina propria, there will be the nerves, the arterioles, the venules, and a new structure called the lacteal, which is essentially a lymphatic capillary. And you might have lymphatic tissue in the form of solitary lymph nodules right up to the surface. They are protecting the surface of the small intestine. Plus, you will see cut sections of these intestinal glands, which don't seem to be connected to the surface, but they'll be looking like cut sections of some tubular structures within this lamina propria region. You won't see this beautiful arterial to venous capillary shift which is happening, but that is what is happening in the core of the villus. The arteriole is splitting up into many fine vessels into a capillary network, which reconstitutes into a venule, which venule goes out in and takes away the absorbed digested food. Extremely important architecture, but you don't get to see it very clearly. You get to see cut sections of various tubular things in the heart or in the core of the villus, and an important structure there is a lacteal, which doesn't stain well, the content of which is the absorbed lipid component of your food. Now, the same thing, when you come to the large intestine, the function has changed. It's going to be only absorption of water and minerals. So we do not have this complicated and elongated, very elongated surface. Instead, we just have epithelial cells which are lining the intestinal glands or crypts, but there is absolutely no villus formation. And this is a very simple lining. There are no complicated lining cells either. There will be a lot of mucus secretion added towards the end, because after dehydration of the food, towards the end of the large intestine, you will have hardened food, or rather hardened contents, which are going to become the feces. And therefore, towards the end of the large intestine, there will be lots of mucus secretion required to lubricate the passage of this semi-solid and hardening feces out. So we will look at that in the next class, but today we will look at how this small intestinal architecture is modified not only in the terms of surface features which are visible to the naked eye, but micro features with each type of cell provided, and subcellular features where each cell gets further modified to perfect and to sophisticate its function. So let's go on to the bigger picture. At this, I think you all can read the lining, the labeling better. So epithelial cells, including a lot of goblet cells. Remember this, we will look at that in detail. Intestinal glands called the crypts of Lieberkuhn, Lieberkuhn. It will come in the next slide. And then you have the lamina propria, which is essentially reticular fibers network. Little bit of connective tissue. It will have a little bit of collagen and elastin also, but mostly reticular fiber network. Muscularis mucosae, circular muscle layer. Myenteric plexus. You remember there was a submucosal plexus here, Meissner's, and there is a myenteric plexus, Auerbach's plexus. And longitudinal muscle layer, serosa, consisting of muscularis externa. Then there is a submucosa. Inside this, I had named the microvilli, capillary networks, nerve, lacteal, vein, and artery in the core of each villus, lymphatic nodule. So let's look at it, how it looks. In the naked eye, if you opened out the tube of the large intestine, you see that we have a plica circularis. Right? These folds are also called the valves of Kirkrig. That's an E. I'm also learning new tricks for distance learning. This will be there in your books. Please look at it. I think it's there in some of my labeling also. Alright, this is an E. Alright, Now, they are not valves. Please remember this. You're looking at it with the naked eye. I'm showing you this because you haven't done the dissection yet. So when you have this 5.5 meters of intestine and you take out the tube, you see that the beginning of it looks a little bigger, thicker walled, and by the time you get to the end of it is thinner walled, and the lumen seems to have become smaller. Here the diameter of it is bigger and the diameter of it becomes smaller. But if you slit it and open it and look at it, this is the longitudinal axis, right, from here to here. Along the horizontal axis, along the circumference of the tube, you see these folds of mucosa, as if the inside is folded upon itself in the form of parallel folds. And these are called the plicae folds circularis because they are along the circumference of the intestine. So if you open it up, each fold is forming like a half shelf. And it is slowing down the passage of the food as it empties after the stomach through the pylorus into the first part, that is the duodenum. The food gets slowed. There's no smooth passage. There is folds of mucosa which are interrupting it. Therefore, it slows down the passage of the food through as it passes through the gut with peristalsis. It was assumed, I think, at some point that in some way they're working as valves, which are manipulating or which are impacting the way the food moves along the gut. So there's no actual valvular action. There is no muscular contraction or anything. But there is the look is that's why they were called the valves of Kerckering in earlier anatomy books. But the correct or the Latin name is plica circularis. Just remember, these are not seen in the microscopy. When you take a small bit of it, you won't see the whole valve, but you might see a full fold in section on top of which are villi. So first level folding, second level folding is a villi, and the third level folding is on the surface of each of the lining cells are microvilli. This increases the absorptive surface exponentially. Several 100 square meters of absorptive surface are available now for rapidly and efficiently absorbing the food which is getting digested as it passes through. For digestion to take place, you must have some digestive enzymes poured into this. The major enzymes are brought in from the liver and pancreatic secretions, but a good amount of enzymes are also added by the intestinal secretions. So some part of the lining will also have to have cells which secrete succus entericus. You have heard this probably from earlier. Their intestinal juice which contains digestive enzymes. So there will be secretory cells, but the large majority will be absorptive cells. So please remember that this is something you see with the naked eye. This is not a microscopic feature, but then the term keeps coming up. Remember, plicae circularis, and you will look for it when we actually do the dissection. This same plica, the same fold, this is the surface of a fold. The plica is actually this fold. So if you look at it under a scanning view or a very low magnification view, what you're looking at with naked eye is this fold, right? And then this fold. If you slice through that and stain it and put it under a microscope, you will see that the whole wall, not the muscle wall, but the submucosa along with everything in it moves into this fold and is covered by the mucosa which is in the form of villi. So these foldings are villi, and this fold is the plica. But you won't be seeing it at this power when you come for identification, because we want you to look at this structure of the villus in much more detail. So for identification you won't see a plica, but if you get a slide for discussion, then you should be able to look at it. If you put it under the lowest magnification, you may be able to make out that the lining, the wall itself has these mucosal folds. So the correct word is mucosal folds, because only the mucosa which is going almost in, but remember submucosa also is going into the core of each of these plica. Muscularis is not participating. The valves are not filled with muscularis externa. Therefore, there is no impact. There is no contraction from muscularis within each of the plicae. So this is from here to here is mucosa, then there's a little submucosa, then there is inner circular, outer longitudinal serosa. From here to here is the wall of the small intestine. This is what the plica look like in a scanning view. Now let's look at this. This is an important slide for you to understand. In the slides, in the stained histology slides, you will not see this much of detail, but you must know that it is there, and you must be ready and prepared to discuss it. So what we had said is this: if this was the surface, if this line was the surface, innermost surface, because this is the lumen, right, and this is the surface. Rising above the surface is the villus, an extension of the inner surface into long, long folds. Dipping below the surface is the intestinal gland or crypt. Now these are called the crypts of Lieberkühn. I'll write that again in the next slide. But you remember that plica circularis is valves of Kürkring, and these intestinal glands were first described by a German called Lieberkühn and named after him. Crypts, because they are hidden. So if your eye is here, you're looking in it, they also look like pits. If you remember, in the last class we saw gastric pits. If you look at the surface of the stomach inside, from the inside, it feels and looks velvety, and it seems to have pits in it. Now, even in the small intestine, you'll get to see much, much smaller pits, but they're very simple tubular glands. They don't have any complications. Simple tubular glands which are dipping in, and again surface, then dipping in, and again surface, then rising up as a valve, and again surface, right? So what we will see is that in the core of each of it, there will be a vascular network. There will be a vein and an artery. So that means the blood vessels have penetrated from the serosa, gone through the two layers of muscle, started breaking up into arterioles and finer branches, then gotten into the submucosa and the core of the villus, and they're giving off branches profusely, which divide into a very rich capillary network. You must remember these are fenestrated capillaries which allow big molecules, macromolecules, to get absorbed into the blood supply. And then they will reconstitute into venules and come out as a vein which will drain away, taking away all this digested food and of course deoxygenated blood, plus of course waste materials. So the lamina propria is here. The muscularis mucosa, if you remember, these three: epithelium, lamina propria, muscularis, are the three constituents of any mucosal surface of the entire digestive tract. That's the plan of the entire GIT. So muscularis in this diagram is not shown, but remember muscularis also goes into the core of the villus, and then beneath that is the submucosa. Submucosa looks a little featureless. There's nothing much going on there, but remember there is a submucosal plexus, a nerve plexus. Doesn't stain very well, but remember that it exists and that it will supply the smooth muscle which also fills up the core of the villus, which means this smooth muscle has come from the muscularis. Right? You may not see that continuity in your slides. And smooth muscle is there. If this smooth muscle contracts, it can shorten and heighten the length of the villus, and it will help to mix up the chyme. By now, the material inside the lumen is called chyme, chyme, and that is the food material, partly digested, all the juices that have been added so far, salivary juices, esophageal secretions, gastric secretions, and now they will land in the duodenum. Duodenal secretions will also be added. All of that together with that semi-liquid, almost entirely liquidified food is called chyme, and it is here in the lumen. And this smooth muscle will allow the chyme to mix well while it is being worked upon by the digestive juices. So these are the structures or the features which are present. Then you have a new thing which was not there in the gut so far, at least not visible, and that is the lymphatic capillary network. So we have a huge network over here. You don't see them. You have lymph nodules which will be seen as solitary lymph nodules. You've seen lymphoid organs where you've seen bunches of nodules in cortexes in the different lymphoid organs that we studied. Here the whole organ is not lymphoid, but it is a lymphoid feature which is very, very important in the function of the small intestine. So you'll see a few thousand lymphocytes, dark purple dots in a ball, oval looking ball, and you will not see these structures which are lymphatic vessels which are interconnected with this, but you may see a long cut section, an LS, through a simple lined, it is actually like a capillary, a lymphatic capillary, but it won't be containing RBCs. It will only be containing mainly absorbed fatty acids and cholesterol, the products of lipid digestion, as well as some lymphocytes and some WBCs. So this lymphatic capillary being at the core of each villus is a very important feature which you need to remember, which is new. And this particular component of your food starts getting digested and absorbed here itself. Then there will be an afferent and an efferent limb, and the efferent lymphatics will go away, taking all this absorbed fat in a different type of lymphatic circulation. We will look at that later. Physiology will teach you that better. But remember the 4 layers: mucosa with lamina propria, muscularis and surface epithelium, submucosa, muscular layer with inner, circular and outer longitudinal, and a thin layer of serosa. This serosa, as you can see, simple squamous, thinly flattened out cells on the outside, because almost the entire small intestine is intraperitoneal, which means it will be wrapped in a layer of peritoneum. So that inner layer, visceral layer of peritoneum will be secretory in nature. It will be secreting a thin serous fluid outside this gut tube, contained in the peritoneal cavity outside the tube. So the function of the serosa will be to secrete that peritoneal fluid. Inside of this, there will be a little connective tissue domain into which the large vessels, nerves, lymphatics, everything are coming in, and that connective tissue will hold together the whole tube and give it its shape. So this is the basic architecture of the villus and the intestinal glands inside the small intestine, all 3 parts of it. So this is a prettier picture just to show you all of those components put together, right? So what we've got is a mucosa from here to here, then you have a submucosa, then you have a muscularis, and then you have a serosa. So you can they put smooth muscle cells here in LS for circular and in TS for longitudinal, and they're showing you the yellow, which is the lacteal and the lymphatic network with bigger lymphatic vessels, and then it will get out and take away the lymph. What are these arrows? Firstly, these are the finger-like projections with as a villi. If you do a cut section, this is how you see it. Outside most is the surface lining, epithelial lining, and in the core of it are all these tubular structures. These tubular structures are the central lacteal, the venule, the arteriole, the little bits of connection between the two. If you can see, the lymphatic capillary is ending blunt. It's not interconnected like a artery vein, red-blue. It is ending blunt. This blunt tip of it, the entire thing is simple squamous epithelium only, but it is able to absorb large fatty molecules through this, and then it will flow out and become bigger and bigger channels and exit the wall of the gut. The other thing they're showing you here, I don't know if you can make out, are this little bunch of lymphocytes, small bunch, and this looks like a big bunch of lymphocytes, just dark purpley staining dots, and they are right under the surface epithelium. This is a very important feature. We will look at that again. It's not been labeled here, so we will look at it again. This is to give you an idea of how these finger-like projections are the villi. This is schematic. When you look at it in a slide, it looks different, but you must remember that this is the architecture. Now these arrows which I was talking about, this is the direction of movement of the cells which are going to replace the surface cells. These cells are shedding off. There's going to be a lot of friction here. So these surface cells are going to rub off and sometimes fall into the lumen and get lost. But always the surface will be healed and reconstructed on a continuous basis. The lining cells of your gut are among the highest turnover cells that you have. Mitosis is happening here at a furious pace compared to the rest of your body. It's an area where continuous cell division is happening to replace the lost cells, just like the epidermis of your skin. So if you have a simple columnar lining, inside the intestinal gland here will be basal cells which will be able to replace all other types of cells, and they will be pushing upwards and getting onto the surface of the villus. Here they'll differentiate. By the time they come here, they differentiate either into a surface absorptive cell or into a goblet cell, which is a secretory cell for mucin. So these little white ones are goblet cells. So the replacement cells will go and take up the position and do the function which they have already differentiated into. When the surface cells are lost, they will just move and cover up the gaps. So the surfaces of the villi are prone to the friction, the mix with the contents continuously on a continuous basis, and therefore this provision is there for rapid cell division, maturation, specialization, and continuous replacement of lost materials. So these are the things I wanted to show in this particular. This is what it will look like. in high magnification if you actually see it. So this is where you see, I don't know if you can make out, this is a capillary lined by a squamous nucleus there, capillary with 1, 2, 3 nuclei. This gap is the lacteal, right? So you have a lacteal, which is essentially a lymphatic capillary with simple flat squames lining it, and the inside will contain no RBCs. Be a very empty looking space because it's essentially fat filled, but you may see a few lymphocytes. You may see a few. Then you have capillaries and lacteals. You have some fibrocytes which are able to secrete the reticular fibers which will keep the core architecture of the villus. We'll see some smooth muscle cells which are extensions of the muscularis mucosa, which has entered into the core of the villus and which can change the dimension, the height and the width of each villus. You may see some emigrating lymphocytes. Is very important. Lymphocytes can squeeze through the lining cells and be almost, this one is in between two columnar cells of the surface epithelium, and they can fall into the lumen sometimes, present right up to the surface, or be present there to see whether there are any foreign proteins or which they can start producing antibodies. So this second level defense mechanism is extremely important as far as the small intestine is concerned. Most of the cells are lining epithelium is simple columnar, which are of an absorptive nature. Few of them, like this empty looking, are the goblet cells. So these are goblet cells where the mucin has been washed off in the processing of the slide, and it doesn't stain with H&E in any case. So you have empty looking ovoid spaces where the nucleus is deep, is near the basement membrane, and the surface is full of its secretion called the mucin. Now wandering cells would mostly be derived from the WBC series, which could be also fibrocytes, which will lay down the fibers. So they are non-specific pluripotent kind of cells. Then you have smooth muscle cell, again to allow the villus to contract. Exfoliating epithelial cells. You see this area is usually disturbed because the cells may be getting lost from there. And a new feature, very important, called the striated border. Now on the surface of these cells, you remember when we saw pseudo-stratified ciliated columnar epithelium in the trachea, in the respiratory epithelium, we could see a fuzzy looking surface even at 100x. Here also we have a fuzzy looking surface, but there it was cilia, which are intracellular organelles. This is not organelles. This is only the surface cytoplasmic membrane thrown into fold. So the complete difference. And the function is also different. There the cilia were specifically made or created to move the mucus sheet away from the lungs and out. Whereas here the surface epithelium has been folded upon itself very, very finely to be able to increase the absorptive surface and to be able to take in as much material as possible. But it looks a little fuzzy. It looks like vertical lines if you look at it at slightly higher power. So they call it a striated border. It looks like there are striae inside it. Even at this magnification, you can see it's very fine parallel lines at right angles to the surface. So this striated border in the structure of a villus is extremely important. This is a diagram, but again you need to remember the villi, same thing, surface epithelium, striated border which is equal to microvilli, lamina propria, smooth muscle fibers. If you look at it in TS, you see there's a central empty looking vessel-like structure, basically capillary-like structure, which is a central lacteal, and maybe you may see one or two others which would be the arteriole and venule, and you would see the surface cells which are simple columnar, all of them. And this one, the orange reddish stained one, is the goblet cells, which means you need a separate stain because goblet cells don't stain with HE. So please see here, it's called periodic acid Schiff stain. You need to put that in to be able to see goblet cells separately. It won't stain the remaining cells, so they look colorless except for the nuclei a little bit. So that particular reagent is required to bring out the goblet cell and to stain the mucin in the goblet cell, which is what has happened here. And okay, so these smaller structures with RBCs in them are the blood capillary. So probably you should name it as lacteal or lymphatic capillary and blood capillary separately, so that you remember. So these would be the blood capillaries, these would be the lymphatic capillary sections. This would be the smooth muscle cells which is supporting the core of the villus. The basement membrane of the epithelium may not be very clear, but in a regular HE stain, the whole cell stains with H and E, so you can make out where the mucosa is separate from its lamina propria. It will show up much better. At medium magnification, actually this is very high magnification, but remember that sometimes we use different stains to bring out the characteristics of different cells, in this case of the goblet cells. So what we have here is a much bigger magnification. The arrow indicates the lumen, right? So this is the surface cell, tall cells, very tall cells, and with a surface striated border. The point was to show you this striated border. I don't know if you can make out it's striated, but it definitely looks darker because the microvilli are so crowded. And this is the core. From here to here is the core of the villus. From here to here is the epithelium, and from here to here is the core of the villus. And inside of that, you have so many features. Here you can see smooth muscle cells. You can't see the cut sections of anything. Again, in this stain, we've used something special to be able to stain the mucin in the goblet cell. The remaining is absorptive columnar epithelium. Here again, this is the lymph. Down the brush border, the striated border is much better seen. So again, this is the lumen. This is again the lumen, seeing the brush border. You are seeing a goblet cell. Again, some different stain must have been used. And this is an absorptive columnar cell. This is to show you the nuclei of the cells forming the core of the villus, and these empty looking spaces are the capillaries, blood and lymphatic capillaries available here. So showing you the serial arrangement of the absorptive cells is very disciplined, systematic, and in between you will have a goblet cell where you don't see the base of it, and you're seeing, in this particular case, you're seeing the surface mucin in that goblet shape. Another stain. Again, this is not for the goblet cells, but it is staining the nuclei more orangish. Homework. Find out what kind of stain will give this kind of picture to the lining of the small intestine. It seems to me the nuclei of everything are looking orangish red, and so are the surface of the intestine. Now, can you make out? This is the striated border, which is the microvilli on the surface of each of these tall columnar cells. The cell is from here to here, very tall, from here to here, with a ovally elongated nucleus, right? And the goblet cell has a nucleus at the base and a bunch of mucin in its surface, which will empty into these gaps between the two columnar cells. This area where you have these surface striated border of microvilli, the mucin has already been secreted and it is covering the surface. And with the mucin covering the microvilli and forming a kind of a layer which is held by it is called a glycocalyx. You might come across this term in your books. Please look at it. This glycocalyx formation on the surface of the microvilli of the lining cells of the small intestine is an important feature. It's called GLYCO, glycocalyx, CALYX. That is the structure of mucin embedded microvilli which are absorbing the contents of the small intestine. Now let's look at each part of the wall of the gut in sequence. So what we'll get to see is that the first part we said is the duodenum, and its function, important function basically, is to receive the gastric contents and start acting upon them, but in an alkaline medium, because small intestinal digestive enzymes work best in an alkaline medium. So we have to neutralize the gastric acidic content. Already that has been done by the pyloric secretions, which had a lot of mucin in it, so that dilutes the acid to a large extent, after which it gets squeezed via the pyloric sphincter into the duodenum. But duodenum must now start secreting a lot of bicarbonate. So we will see that its lining cells will be adding intestinal juice in the form of more amount of bicarbonate. We will look at that. So what we need to observe here in a low power or a scanning view is that from here, lumen, to here is mucosa. Then you have a submucosa. Then you have muscularis, which isn't much, and a little bit of serosa. This doesn't even look serous, right? We are not able to see a simple squamous epithelium here. This is because most of the duodenum is retroperitoneum. It's not covered by the peritoneum. So then you should call this the adventitia. More correctly, the duodenal wall, external wall, does not have a serosa but has an adventitia. That's very important. But in your slide, in your identification, you may not see the external wall because we'll mainly show you the villi. So when we show you the villi, what you need to look at is the look of the villi. They are somewhat broad, right? And many of them are blunt. They're not looking very sharp or pointed. This one is looking sharp, but if you look at jejunum, you'll see there's much sharper tips are longer kind of villi. So this is the look of duodenal villi. The surface has got simple columnar epithelium and a lot of goblet cells, those little round white empty dots, right? Those are the goblet cells and surface epithelium. You can't make out microvilli or striated border at this magnification, don't try. Just look at this folding nature, and that should tell you it is small intestine. Any part of the gut which has this multiple folding nature of mucosa has to be small intestine. Once you have small intestine, this must be the surface. Above it is the villus. Below it is the intestinal gland or the crypt of Lieberkühn, right? So these are the intestinal glands, simple tubular glands going down into the lamina propria. Again lined with this almost the same epithelium, but there are other cells here which you can't make out at this low magnification. So in the lamina propria, can you see a whole lot of black dots? There will be a lot of free lymphocytes floating around. And this is a bunch of lymphocytes right under the surface. We will see it at a higher magnification. Then you have the muscularis, and then you have a submucosa, and you have another bunch of glands here. Whole lot of secretory mechanisms here. And this is a feature which differentiates duodenum from the other two parts of the small intestine. These are called the Brunner's glands, submucosal, mucosal secreting glands, not only mucin but also bicarbonate ions. That is the important thing. These glands are here and they add an important bicarbonate component so that the contents in the lumen now become alkaline and can be acted upon by the digestive enzymes. So, mucosa with broad, wider villi, crypts of Lieberkühn or intestinal glands, and then submucosa with Brunner's glands. Then muscularis, it doesn't look very, you can't see the longitudinal layer separate from the circular layer. There's more circular and a little bit of longitudinal. And then you have an adventitia with some vessels here. You don't see the Meissner's or Auerbach's nerve plexuses clearly at a low magnification. The point to remember is it won't have a serosa and it will have this particular shape of villi and it will have Brunner's glands, submucosal, mucin secreting glands. Structure of the duodenum. Now, this is a painting. Again, this is a diagrammatic view. The last one was an actual microgram. So what we are going to see is that you have surface villi which are looking a little wider. You have wider intervillous spaces, more gap between the villi. You have goblet cells. You have duodenal glands which are stretching into the lamina propria. This is all supposed to be submucosa, right? From submucosa it is going off and you may even see them getting into the duodenal folds. Then you may see lymphatic nodules which are coming right under the surface. A lymphatic nodule is very sharply demarcated because of its dark color, lymphocyte stain purple. So if you are seeing these purple dots right under the surface, then it is a lymphatic nodule. And you may or may not see the myenteric nerve plexus. You must remember the villus has a lining epithelium, a lamina propria. Then apart from the villus, there are intestinal glands. There is a muscularis mucosa with smooth muscle fibers. There are lacteals. I don't know if you can make out. You're supposed to draw this. A thin capillary-like structure in the core of at least some of the villi. You please draw. In the fatter villi, you can draw a bigger space. This is not a lacteal because it is not lined by simply. This is a cross section of a villus at a different angle. So don't get confused with that. In any one villus, you won't get a pure LS longitudinal section because they are finger-like. They are not straight standing structures. So when they are wavy, the cut section will not go through and through the whole length of them. Some cut sections will look like this. Don't worry about it. Wherever you see one long continuity, you draw the lacteal in the core of it, you'll be able to see it in some of the villi, an empty looking space in the core. Then muscularis mucosa, duodenal glands in submucosa, very important. You see mucin secreting, therefore pale staining. They do not stain like a what should we say, like your salivary gland or like your pancreas. They stain like a where esophageal glands also were mucin secreting, right? So duodenal glands in the submucosa have to be drawn as cut sections of duct-like structures which will be secreting their bicarbonate-rich mucin into the lumen. Then muscularis externa is inner circular, outer longitudinal. Serosa is the wrong word, please call it adventitia, tunica adventitia. This is more of a photograph, very nice one actually, where you have empty looking spaces in the core of a villus which are the lacteals. All the rest are the same, what I have described before, but it's at a higher magnification. When you are shown at this magnification, the field is not big enough to see all 4 layers of the gut. So your identification will only depend on the inner 2 layers. So in the mucosa you are seeing villi, you are seeing intestinal glands, right? You are seeing intestinal glands. So you are seeing villus up and intestinal gland down. Inside the villus you are seeing an empty looking space. Sometimes you are seeing one empty looking space, and I don't know if you can make out anything feathery striated. If you can't, it's all right. And after the muscularis, you're seeing lots of glands in the submucosa. This has to be duodenum. If you're seeing submucosal mucin secreting glands, then this has to be duodenum, right? You're seeing villi, so it is small intestine anyway. High powered view, very nice staining. You can see this surface dark lining is because of those microvilli forming the striated border. The goblet cells are so prominent. In the core of the villus, sometimes you can see a space, not very clear. In none of these can we see a lacteal very clearly. But the dipping ones, these are the intestinal glands. Wait. These are the intestinal glands. Right? They also seem to have some goblet cells, but they mostly don't. They usually have other secretory cells. We will come to that. This again has a special staining for the goblet cells. It seems to be green. You want to do a second homework and find out what stain it is, just to show you the high power view of what the villi look like. Jejunum. Now this is important to differentiate duodenum from jejunum, from ileum. So the new feature in the jejunum is that the villi are taller, thinner. They are described as feathery, feather shaped, right? They have the same goblet cells, same structures as before. There are some adipocytes. This will definitely have a serosa. Jejunum, entire jejunum is intraperitoneal, so it will have outside a simple squamous lining of serosa. It will have more visibility of lymphatic nodules. As we go down from duodenum to jejunum to ileum, the solitary nodules of lymphocytes become more and more aggregated and become very visible and very dominant by the time you get to ileum. Whereas the villi from duodenum, broad and medium villi, to jejunum, tall, thin, numerous villi, to ileum, shorter, sparse villi. That's how the changes happen. You will still see intervillus spaces, you will still see crypts or intestinal glands. And here you see the submucosa has no glands at all. So jejunum has no Brunner's glands. So absence of glands in the submucosa is the way to differentiate duodenum from jejunum. Right? So what are we seeing at the base of these intestinal glands? Important to see the cells. I told you there will be a lot of mitosis. So you will see mitotic cells, you will see goblet cells, you will see Paneth cells, where you have supranuclear pink staining granules. These are the ones which are secreting the intestinal juice. These are the enzyme secreting cells called Paneth cells. Then you have enteroendocrine cells which don't stain well with He. They are argentaffin or chromaffin cells, remember? So they're called enterochromaffin series or argentaffin cells, which will secrete neuroendocrine substances, hormonal substances, both local hormones which aid in digestion as well as other hormones which are secreted into which fall into the circulation and go and act elsewhere. So just remember that they also exist. They don't show very well in this stain. Lots of mitotic cells and No absorptive cells in the depth of the gland. The glands are not for absorption, the glands are for secretion. The surface cells only will have absorptive cells with microvilli. So this is the deep structure. This is true not only in jejunum but also in duodenum and in ileum. The kinds of cells available: mitotic cells, goblet cells, Paneth cells, enteroendocrine cells, are all parts of the entire small intestinal lining. If you look at the histology, these villi are also looking little broad, but they are described as thin, feathery, tall villi. The glands are there with the 4 types of cells, and simple columnar epithelium on the surface interspersed with goblet cells, muscularis mucosa and submucosa. So the look of a jejunum. In the ileum, this is where the ileum is. The ileum ends in the cecum, and never mind this one. But this is to show you that this is the plica, right? This is a whole fold. These are the villi on the surface of the plica. And here should have been the glands, but instead you have 1, 2, 3, 4, 5, 6, I don't know, 10 or 15 lymphatic nodules just under the surface. That entire lamina propria is taken up by these lymphatic nodules. Actually, not only lamina propria, but almost entire submucosa also. Large aggregations, lakhs or millions of lymphocytes together right under the surface. Each of these, if you look at it, you can see it through the naked eye. If you're looking at it from here, if you open out the ileum and look at it, that area will not show this velvety look of the villi, but it will look a little flatter, right? When you open it out, it may be anything from 2 to 10 mm, that big, visible to the naked eye. That it looks like a patch on an otherwise velvety surface. Therefore, they were called Peyer's patches, named by Dr. Peyer, who first described them. They are very important. They denote where the aggregated lymphoid follicle is available under the surface. So, in this distal portion of the small intestine, you have the serosa, muscularis, submucosa, and mucosa. But the aggregation of lymphoid tissue, called Peyer's patches, are found in the lamina propria, just beneath the surface epithelium. All these lymphocytes are available to whatever is in the lumen. They will interact with the external environment. So that provision is very, very important component for the ileum part of the small intestine. This is what it will look like in a diagram. Aggregated lymphoid follicle. Best thing to call it is not Peyer's patches, because Peyer's patches is a gross anatomy term. You're actually looking at a patch on the inner surface of the opened out gut. Better histological word is aggregated lymphoid follicle with germinal centers, with lymphocytes going right up to the surface. Everything else is as I have described before, and the serosa is the visceral peritoneum, which we have talked about. The villi, you may see some what looks like floating villi, but it's actually the finger-like process which has got cut, and you're not seeing the full length of it. So you may see many villi in continuity like this with the intestinal gland, many villi which are just looking like floating cut sections. Whatever it is, they are all villi only. The depth ones cut sections are intestinal glands. And the aggregated lymphoid follicle is a very, very important feature of ileum. Submucosa again has no Brunner's glands, no other glands. Lymphoid follicles are available. In a high power view, again from outside, this is the serosa, longitudinal muscle, circular muscle, submucosa with a core going in, and a whole bunch of lymphoid tissue. You should be able to recognize lymphoid tissue wherever you see it. And it's going right up to the base of the villus and the intestinal gland. From here, these are the villi. And you're looking at them in cut sections. These are all villi. And this should have been an intestinal gland, but it's right, instead of the gland dipping in, there's a whole lot of lymphoids in a full lymph. This is one nodule, this is one nodule, this is one nodule. So many nodules right under the surface. This is characteristic of ileum. So this brings us to the end of this class. Differentiating these 3 slides is a little challenging. You remember that there are 4 layers to the walls of the gut. You look for the features of the villi. Look for the presence of Brunner's glands in the duodenum, the presence of aggregated lymphoid follicles in the ileum, and jejunum tall feathery villi. These 3 points should help you to differentiate. So thank you for today. Please revise this within your histology atlases. If there's anything I missed, if there are any questions, please share it in the chat. I'll see you again in the next class. Bye.